Before continuing, I would like to talk a little bit about the nature of a signal transduction pathway. As we said, a signal comes into the cell. A number of steps occur within the cell that lead to the cell formulating the correct response to the signal it received. Now, what we're interested in this section is what is the nature of the things happening here. We're not really interested in the nature of the response because the response will be different by different cell types, as we will discuss later. What we're more interested in is the molecular mechanism that is occurring once the signal is received and the things that are happening within the cell which eventually lead to the response. So a signal transduction pathway always starts with a receptor that will bind the signaling molecule. Then you have a number of players within the pathway that are in the inactive position. So we'll put 4 in here. A, B, C, and D. And these are in the inactive state. So red is for inactive. Now once the signaling molecule has been bound to the receptor becomes activated. Okay? And that will lead to A becoming activated. The receptor itself is involved in the activation step of A. So green is for active. Now A, activated A, will be involved in activating B. Active B will be involved in activating C. Activated C will be involved in activating D, and we're going to say that D is our effector protein. And D will go on to initiate the response. And we'll put R for the response. Now, for each one of these steps, there is an inactivation process that is always active. Now the arrow for the inactivation process is smaller than the arrow for the activation process. So once the activation process is started, it will outpace the inactivation process. So you get a buildup of these active proteins within the cell. Now, once the receptor is unoccupied, meaning that the signaling molecule is no longer bound, the receptor is no longer active, well then this arrow disappears. And the only process that is active for A is the process which is inactivating A. So active A concentrations go down. Well, if there's no more active A, then the activation process for B is gone, so the only process left is the process which is taking B from the active state to the inactive state, so the concentrations of active B decline. Now, if there's no more active B, well, the activation step for C is gone, so the only process that is left is the inactivation step for C, so the concentrations of active C go down. No more active C, no more activation of D. The only process that is active is the process which is taking active D and taking it back to inactive D, so D is gone. And thus this is gone, and there is no response. Right? So this is how a signal transduction pathway is turned on and turned off. Now let's rewrite the turning on process again. So how this is written to make life a little bit easier, instead of having to write all this out, is signaling molecule activates receptor, which activates A. A goes on to activate B. B goes on to activate C. C goes on to activate D. And D will lead to the response. So the arrows that are being shown here, 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 and here, these are activation arrows. These are not chemical reaction arrows. So when you see that arrow, you know that one thing is activating the next, and you should be aware of what is happening, that this is actually what is happening. Something like this. Okay? Now, there are conserved pathways in all cells, and these are conserved across species too, animals. So what they have in common is perhaps the signaling molecule, the receptor for that signaling molecule perhaps, and the first few steps. But then the effector protein might be different between them. So this part is conserved. Everything downstream from C is going to be a little bit different in different cells, meaning that in one cell the effector protein could be one thing, and in another cell the effector protein could be something different. And that's basically based on differentiation, meaning that certain effector proteins are silenced in one type of cell where they're active in another. In some cases, the receptor and the signaling molecule might be different, but essentially this part is conserved, meaning that this mechanism within the cell, this A to B to C, these activation steps, are conserved across different cell types. So for example, we can have cell 1 and cell 2. And these are two cells that are specialized, and they respond differently to the same signal. So here's my signaling molecule, and we'll just write signal or stimulus. Same signal. And you have the receptor. That is the same. Being activated, we'll just put REC for both, for receptor. And then you have the steps in between that are the same between them. But the two start to diverge at this point. For example, cell 1 will activate D, which leads to a response where, in this case, this will activate E, which will lead to a different response. The responses could be completely different from each other. Okay? So this whole part here is basically conserved between the two cells. But everything downstream from this part is going to be different, including the response. So in this section, we're just going to be talking about these conserved, a few of them, not all of them, pathways that exist, that are important to biology. There are common signaling pathways that are found across different animal cell populations. These conserved pathways are equivalent in identity of proteins within the pathway and the mode of activation. The activation of these conserved pathways in different cell types will lead to different responses. A large majority of pathways from receptor to intracellular components are conserved, but the effector proteins that are activated in the end are different across varied cell populations. Thus, the activation of these pathways will lead to different responses depending on the effector proteins being expressed. Note, for the remainder of this section, we will cover some of these conserved pathways and their particular features. Alright, and that's it for this part. And we will continue later. Thank you guys.